Hello, my name is Steve Trim. I am the Chief Scientific Officer of Venomtech Limited and the Chair of the Veterinary Invertebrate Society. Thank you for taking the time to look at our poster on thermal nociception in large theraphosid spiders using the Hargreaves apparatus. This was a collaboration between ourselves, Transpharmation, Canterbury Christ Church University and other members of the Veterinary Invertebrate Society. For those of you not aware, theraphosid spiders are the technical term for tarantulas, so the big hairy spiders, sort of 30 to 40 grams, and therefore I've put a warning out there for anybody with a nervous disposition for these animals are featuring in this presentation. Pain processing in invertebrates is quite poorly understood. As we all know, it's a sensory and emotional experience, and we know very little about the sensory experience and let alone the emotional side of things in invertebrates. Most work's done with small model species such as C. elegans and Drosophila, and they really struggle to be used in standard rodent techniques. Therefore, we hypothesized that larger theraphosid spiders such as tarantulas would actually be more suitable for rodent equipment and therefore help us understand a bit more about invertebrate pain processing and wondering if this could actually lead on to further use. A fundamental hypothesis for this study is that all animals have some form of a nociceptive response just purely for tissue protection and defense. We would therefore thought that theraphosids would be no different. So we went to look at what are the baseline responses, because this had never been done before. We didn't know if the Hargreaves equipment would actually detect a foot withdrawal from a non-vertebrate. Also then to look at the temperature, and then as we went on, how do these responses change with simple sensitization. And here we have our Hargreaves apparatus set up, in this case on a downflow table, because theraphosid spiders, particularly from the Americas, actually produce these urticating setae or hairs from the abdomen that can be really irritating and therefore a danger to the staff. So all animal manipulations are used with the downflow table, which then turned off for acclimatisation period, mostly 5 minutes, but we have the 5 to 10 minute window to make sure that the spiders were at rest and ready for a accurate test, but we didn't want to leave them too long because they will cool down and that will reduce their motor responses. After the acclimatization period, we're able to position the stimulator on the plantar surface of the foot as it's in contact with the glass plate in much the same way as you would with a rodent, and push the stimulator button, and we were really relieved first test to actually see the infrared stimulator did detect the foot withdrawal of the spider reproducibly, and we were then able to take this method forward in our experiments. In looking at this response was the first time we'd seen this, so we actually used a calibrated thermal resistor to measure the withdrawal temperature and found it to be within 50 degrees, which is exactly as we would expected for mammals. Was great to see it in invertebrates too. We tested a wide range of genera, and the Grammostola genera were not only the most useful with regards to normal distribution of responses, but also compared to Pteronachilus, they were much less agile and much easier to test. Once we had selected the species and got good baseline numbers, we then moved over to actually looking at very simple sensitization. The first was just a thermal stimulation from the Hargreaves apparatus 30 seconds later from a first stimulation, and we saw then a reduction in the latency showing that the first stimulation had temporarily sensitized the foot. To then look at injury model, we anaesthetized the spiders with a rising concentration of carbon dioxide, used a 0.4mm needle to cause a needle prick in the plantar surface of the tarsus, let this heal for 48 hours, also to make sure that the full recovery from the anesthesia, and then proceeded to assess the withdrawal latency over time, comparing it back to the uninjured. And what we saw here, that over the study of 83 days, it took 52 days to actually return to a ratio of 1, i.e. no difference from the uninjured, and this was much longer than expected. As you can see from the data presented in this poster, not only do the theraphosid spiders have a suitable withdrawal response to thermal stimulation, but also that the Hargreaves instrument in its standard setup can detect this foot withdrawal. We were really pleased to see that thermal stimulation was around 50 degrees and that repeated stimulation actually reduces withdrawal latency, as you would expect for sensitization. What was quite a surprise is that the time taken for a needle stick injury to return to normal levels was much longer than we anticipated. And we're looking forward to further work on studying analgesics and further understanding pain processing in these big impressive spiders. Thank you very much for listening.